Okay, well, welcome everybody. It's my great pleasure to introduce today's speaker, um, Dr. Andrew Lees, the founder and scientific director of Fina BioSolutions. Fina BioSolutions is a company based in Rockville, Maryland, that is promoting the development of affordable conjugate vaccines by making technology available for emerging market vaccine manufacturers. So a little background on Andrew. Andrew is a proud native Bostonian and originally got his undergrad Bachelor of Science degree in chemistry from Harvey Mudd College, where he returned to the other side of the continent to Johns Hopkins University, where he got a PhD degree in biophysics. So I noticed in Andrew's bio that he has many prestigious honors, but the one that I was most struck by was the fact that he appeared on the cover of Baltimore Magazine as one of 84 people to watch in '84. And that was, I think, less to do with his biophysics, but more to do with the fact that he was a leading magician in the Baltimore area. So I'm told that if this were a real seminar and not a webinar, Andrew would do a few magic tricks for us, but this webinar format kind of cramps his style, so I guess we'll have to wait to see Andrew in person before he shows us some of his magic tricks at least. So before he started Fina BioSolutions in 2006, Andrew was also director of vaccine development at Virion Systems and Biosynexis. Currently, he's also associate professor at University of Maryland School of Medicine, Center for Vaccine Development, and adjunct professor at Uniform Services University at University of Toledo. Andrew has authored over 70 publications and 25 patents, mainly in the area of conjugate vaccines, including development of efficient linking chemistry that's widely used for preparing conjugate vaccines for bugs like Streptococcus pneumoniae and meningococcal disease. So Andrew's talk today is entitled Simplifying Conjugation, Ready to Conjugate CRM197 carrier protein, and I'm very excited to hear more about what he has to say about vaccine development. So thank you for joining us today, Andrew. Welcome.
Thank you, Ken, and thank you to the Canadian GlycoNet for inviting me. It's through Ken that I got introduced to the GlycoNet group, and I'm very pleased to be presenting this seminar today. I do usually start with a trick. Today's first trick is to make the economy disappear. Done. Good morning or good afternoon, depending on where you are. This talk will have a slightly different focus than the one in the title, and as well as the one in the title on this slide. I'll be giving a brief overview of conjugate vaccines, and before I move into the protein itself. First, a little bit about Fina BioSolutions. As Ken said, I founded it in 2006 based on my expertise in conjugate chemistry, and our mission is to promote the development of conjugate vaccines. We currently have a staff of 8 people and a very nicely equipped lab for the synthesis and characterization of conjugate vaccines. Our core areas are in conjugate vaccine chemistry, and more recently in protein expression and purification, which is the origin of the CRM197 carrier protein. We also have another group, which is a little bit more commercial, focusing on modified dextrans, and if you're interested in that, you can learn more on our website. So first, why conjugate vaccines? Many bacteria that cause significant disease are covered with a capsule polysaccharide. This fuzzy coating serves a number of purposes. It's a virulence factor, and it also serves to deflect the immune response. In the case of strep pneumonia, there are roughly 100 different types of capsules. And if you make antibodies to one, they're not protective against another strain. So they're highly specific, and in fact, the serotypes are determined by their carbohydrate. Now, polysaccharide antigens are T-cell independent. They show no affinity maturation. There's no memory response, no class switching, and most importantly, they are very poorly immunogenic in infants. So you can't easily raise antibodies to the polysaccharide. When you covalently link a protein to it, the so-called carrier protein, it converts the polysaccharide antigen into a T-cell dependent antigen, and now the polysaccharide shows affinity maturation in the response, a memory response, class switching, and immunogenic in infants. Just as an example of a conjugate vaccine in action, in sub-Saharan Africa, there is what's called the meningitis belt, and they have periodic epidemics of meningitis. This shows the waves of group A meningitis over the last 70 years or so. On the right, where you see the flattening of it, that is the introduction of the meningitis A polysaccharide conjugate vaccine called Menafric, produced by the Serum Institute of India. And you can see that the epidemic essentially goes away. The disease disappears. Not shown on this slide are some of the other serotypes which are still bouncing around, and in a few years there'll be a pentavalent meningitis vaccine introduced in Africa. Another type of conjugate vaccines involve haptens. These are small antigens that are poorly immunogenic. Examples would be chemical moieties, peptides, and of interest to this group, carbohydrates or oligosaccharides. When you chemically link these haptens to a carrier protein, the protein provides T and B cell epitopes and creates an immunogen, thereby permitting an immune response against the hapten. And it's principally against these that I'll be discussing. I've been in the field of conjugate vaccines for essentially my entire scientific career. And just to give you an indication of some of the many areas where conjugate vaccines have relevance, these are some of the diseases and indications that I've worked on over my career. I got my start in this field through the accidental development of a chemical called CDAP, which is one of the most efficient reagents used for the activation of polysaccharides. It is, in essence, a cyanylating reagent, very similar in action to cyanogen bromide, but it has a great number of advantages over cyanogen bromide, and it could be the subject of another talk. It is used in a number of different, both licensed vaccines and vaccines under development. Most recently, the Serum Institute of India, which is one of the largest vaccine companies in the world, had their 10-valent conjugate pneumocil, WHO pre-qualified. And for the poorest countries, the cost will be about 1/3 less than the previous vaccines. I mentioned the meningitis vaccine. For the Serum Institute, I developed a related chemistry which permitted them to circumvent some blocking IP. And this chemistry will be used for all 5 serotypes and is expected to be licensed in around 2022. Returning to carrier proteins, there are For academic or research carrier proteins, perhaps the two most common ones are bovine serum albumin and keyhole limpid hemocyanin, or KLH. Licensed ones include tetanus toxoid and diphtheria toxoid, but the academic ones are really not suitable for clinical use. KLH is available for clinical use, but as a natural product obtained from sea snails, it's difficult to commercialize. The toxoids are difficult to impossible to obtain. Generally, you need to be connected to big pharma in order to obtain them, and they tend to be non-homogeneous. Just as an example of tetanus toxoid, this is the size exclusion chromatography chromatogram of a tetanus toxoid preparation, and you can see that only about a little more than a third of it is actually the tetanus. Most of it is sort of aggregated junk and other things. There was a paper that showed there were over 1,000 different peptides in the toxoid. There's a poster on this. We looked at a number of different tetanus toxoids from a number of different sources. You can find that on our website if it's of interest. The licensed carrier protein that I haven't mentioned yet is the CRM197 or CRM197, which is a genetically detoxified diphtheria toxin. It has a single mutation. Glycine 52 is mutated. This protein is smaller than tetanus. Tetanus is about 150 K. This is about 60 kilodaltons. It has two subunits, and it is very widely used in conjugate vaccines. Notably, it's used in Pfizer's strep pneumo vaccine, which is probably the world's biggest selling conjugate vaccine for strep pneumonia. Unlike the toxoids, which are treated with formaldehyde, CRM is as a recombinant protein. It has its full complement of lysines. So out of a total of almost 40, about 20 are surface accessible, and 7 of these are most exposed. CRM also has 2 disulfide bonds. In its natural action, diphtheria toxin is cleaved or nicked, and it's important in making CRM that the protein remain intact. And so you'll see one of the assays or for intactness is looking at this nicking. The diphtheria toxin is taken up in cells through a binding receptor. It binds and then inserts itself into the membrane, ends up in lysosomes, and in the low pH environment, it turns itself sort of inside out. So as a result, CRM has rather poor solubility in acid and tends to start precipitating or aggregating at lower pH. Until recently, CRM was only available in what's called native form from Corynebacter diphtherium. Yields are fairly low, and it's a fairly tricky bacteria to grow. In around 2000, the company Phoenix started producing CRM in their proprietary strain, Pseudomonas fluorescens. I was part of this project while I was working with the Serum Institute of India, and this CRM is used in their strep pneumonia vaccine. It's also been expressed in E. coli, in periplasma or inclusion bodies, generally at lower yield or more effort to do refolding. About 5 years ago, we were invited to work with PATH and to see if we could express CRM in E. coli using some of the methods that we had developed in-house. and we succeeded. We call this protein as our trade name EcoCRM for E. coli CRM. The strain that we developed has an oxidative intracellular environment, and unlike other similar strains of E. coli, like the New England Bio Shuffle, this strain grows like gangbusters. We get very high yields, upwards of 2 grams per liter of purified protein. The protein is expressed as a intracellular, properly folded, soluble protein. It is, so it's quite remarkable. And when we set out to do this, our goal was somewhere between 0.5 gram and 1 gram per liter. So we have far exceeded the initial goal that we set out with when we began working with PATH. We have a simple manufacturing process. It's basic E. coli fermentation, lysis, 2 ion exchange steps, tangential flow, and then the final formulation. We've been issued several patents, US patents and other patents around the world, both for the production of CRM and related proteins in this E. coli strain. I'll discuss how we ended up working towards GMP CRM for clinical use. But this is from what's called an engineering run. These are the runs which are done, sort of practice runs before you do your actual GMP run, and gives an indication of the quality of the protein. You can see that by size exclusion, the figure on the left, instead of that very messy chromatogram, you now see is a nice single sharp peak. All the necessary qualities like host cell protein, nucleic acid, endotoxin, and the nicking that I mentioned are all quite low. So it is, we're expecting in the next month or so, depending on how the coronavirus treats our CMO, to begin the first run. We currently have it available for research use. PATH funded in order to evaluate and show that our CRM was comparable to CRM from other sources. They funded a analytical study at David Volkin's lab in Kansas. And this is just one little piece of it showing the higher order structure. And I don't really want to get into all of the details, but what these show is that the structure, the 3-dimensional higher order structure of the protein is comparable in both native and with the CRM produced by Phoenix. We also did a collaboration with a company called Redshift Bio. They have a device they call the AQS3 Pro, and it does a sort of differential IR scans with a high-powered laser, and allows you to using their software, allows you to determine the various structure forms. And you can see in this comparison, the beta turn, alpha helix, unordered, and beta sheet are very similar between the Phoenix CRM and our EcoCRM. And a little note, I'll be giving a webinar with a lot more details on our collaboration together and the work we did on May 28th. This instrument was also used to help develop our formulation buffer, and I'll be discussing that at that time. CRM is normally in pneumo vaccines directly conjugated to the polysaccharide, and this is an example of EcoCRM compare as the carrier protein on pneumococcal polysaccharides for serotypes which are included in Prevnar and one that is not. And you can see that red, which is the Fina BioCRM, those conjugates made with our CRM induce similar responses to ones made with the so-called native or Corynebacter CRM. I'll now move into the topic of our what we call ready to conjugate proteins. Because we're in the business of, we're also in the service business, and we make conjugates for various people as a service as well as for collaborators. We began preparing pre-labeled proteins with with linking groups in order to make our life easier. And it occurred to me that this would be a very useful product for both the research and the vaccine world. So the first group that we did is the maleimide, and this is certainly the most popular one. It reacts with thiols. We also have proteins labeled with bromoacetate, similarly thiol reactive. We have proteins with the squarate ester, which are both amine and thiol reactive. And we've also made these with click reagents. And currently, we have CRM, a tetanus toxin fragment, tetanus toxin heavy chain fragment C, as well as BSA available. So we prefer to use maleimide as opposed to bromoacetate, but as I mentioned, they have very similar, but they both react with thiol groups. The bromoacetate group reacts at pH 9. Maleimide groups react at a more neutral pH to give a covalent linkage of the antigen onto the protein. Comparing the chemistries for the two, first the CRM maleimide and the CRM bromoacetate have similar number of reactive groups. The maleimide is by itself is pH sensitive, so it needs to be, if you leave it around, it will start to hydrolyze. In contrast, the bromoacetate is pretty stable. It is slightly light sensitive, so you don't want to leave it out in bright lights. The maleimide group, as you saw in the previous slide, is a much larger and bulkier group. So you tend to make antibodies against the linker, whereas the bromoacetate is a much smaller, sort of more natural amino acid-like linker. This can be an important criteria if you have a very short antigen or a small peptide or small glycan, because you may tend to get more of a dominant response against the linker rather than your antigen. The maleimide reaction is much faster than the reaction with bromoacetate, and as I mentioned, recurs at neutral pH. And since you're at a lower pH, the issue of thiol oxidation is less of an issue, whereas if you're working at the pH 9, you need to be more careful about preventing, keeping your solutions deoxygenated. If you're making this isn't particularly relevant for vaccines, but for ADCs, antibody drug conjugates, in the serum with the antibody drug conjugates around for a while, maleimides can be slightly reversible. The bromoacetyl linkage is not. I'm not going to go through this slide in detail, but the one on the bottom, SMCC, is very popular among many groups. I don't like it because although it is a longer and more rigid linker, it's bulkier and much more immunogenic than the one at the top, which is my favorite, which goes by the initials GMBS. And this is the maleimide reagent that we use. The NHS ester, which is on the left, is amine reactive. So we're labeling some of those, the lysine groups, on the CRM when we prepare this product. Because we want this to be a stable buffer, a stable formulation, the formulation buffer was quite critical. We provided in a PBS with EDTA and sucrose at pH 6.8. And the reasons for this selection, we have a low buffer concentration since PBS is 10 millimolar. This allows the pH to be easily readjusted. It's at pH 6.8. I mentioned that the maleimide is easily hydrolyzed. It's more stable in acid. So we wanted to keep the pH as low as possible. And then in contrast, the creme is not stable in acid, so we needed to keep the pH higher. And so we selected pH 6.8 as the optimum. We've found that creme is more stable in low ionic strength. We include EDTA, not so much that it's critical for the creme itself, but that way when you add in your thiol group, the EDTA serves to minimize oxidation of the thiol group by keeping down, by binding up heavy metals that promote oxidation. Finally, we have 5% sucrose as a cryoprotectant for freezing. If you don't include it in it, then your creme aggregates. So it's currently a frozen provided as a liquid formulation. I was just about ready to buy a shelf lyophilizer when the shutdown occurred, so I had to put that off and hope that I'll have the money later on to purchase that. So it's important that the creme remains stable as a product. We've done freeze studies, so we've done up to 5 freeze-thaw cycles, and so this means that when you receive the creme, if you're using a larger quantity, you can thaw it, aliquot it into smaller containers so that you have single-use volumes and you don't have to buy small amounts. Just in the highest pH that I recommend using the creme maleimide is around pH 7.4. On the right, you see a figure just showing the stability of the maleimide group over a 3-hour period. You can see at this pH at room temperature, about somewhere around not quite less than 10% has been consumed. Since you're using this you'll normally be doing this in the cold. The hydrolysis will be slower. And since you're normally going to be adding the thiol right away, in fact, the reaction would be occurring, so there'd be even less hydrolysis would be less of a concern. I'll say the little bit of work that we've done with the bromoacetate over the 3 hours, it's basically flat. We have not noticed any change in it's much more stable group. One of the nice things about creme versus tetanus toxoid or KLH, creme is a nice, well-defined protein, and that means you can do mass spec on it. So on the upper slope, graph, mass spec is the creme itself. The lower one is creme maleimide, and you can use this difference in the molecular weights to estimate how many maleimide groups on it. This gave 18 groups. When we provide our number, we actually do an assay for active maleimide groups. So it's possible that during all of the preparation and synthesis, 1 or 2 of these may hydrolyze. When we give a number for how many groups are on there, we mean how many are still able to react with thiol groups. Since we don't have easy access to a suitable mass spec, the easiest way to characterize the conjugates is by SDS page, and using a scan to quantitate the difference in molecular weights. You can see that there's not much of a shift for the creme maleimide, reflecting its low molecular weight nature, and the conjugate clearly moved to a higher molecular weight. Again, you can do mass spec. You can actually see how many peptides are on there if you have a suitable mass spec. And this work was done, as I indicated in the previous slide, by Dr. Isles at UMass Amherst, or I'm sorry, Worcester. Similar to creme maleimide, You can do identify how many peptides are on there using STS page. These two STS page show the same peptide coupled using two different chemistries, and you can see that roughly the same number of peptides were coupled to each, but the bromoacetate one required more attention to eliminating oxygen. So now move on to some examples. The first one is part of how I ended up speaking to you all. This is work from University of Calgary, Dr. Ling and Dr. Ng's groups with a tumor-associated glycan antigen. And the immunological data was provided by the Tanha group from Dr. Rosati. This is about a ganglioside and GD3, GD2. And I'll say I only learned about what this actually was in the last few days. But this ganglioside is tumor-associated and overexpressed in a number of tumors. It is normally O-acetylated. The group synthesis, and this target, this particular ganglioside is a target for the only monoclonal antibody treatment where the antigen is a carbohydrate, so-called unituxin. But it was found that there are a number of neuropathies caused by this antibody. And so as a result of this, the Calgary group went and synthesized a new antigen, very similar to the one that I showed you before, but substituting an N-acetyl group for the O-acetyl group. The N-acetyl group being much more stable. They put a linker onto it, a PEG linker, and a thiol group on the end for conjugation. And I began working with Ken through his interest in our CRM, and we began providing them with the CRM maleimide product. And they formed these, the CRM conjugates, characterized these by mass spec, determined that there were about 6 of these from the difference in molecular weight, about 6 glycans per CRM. They immunized llamas multiple times, and the black curve is the final serum dilution and the anti-glycan response. So you can see that it made a very nice antibody response to the glycan. So just to be current, this came from the April Fool's Day edition of Genetic Engineering News, Understanding Glycans in COVID-19 Drug Design. And this group in San Diego, I'm not sure how they identify all of the different carbohydrates, but you can see at the bottom are the carbohydrates, not the bottom, the carbohydrates that are involved in the binding sites to the ACE2 receptor. So hopefully somebody will be working on this, and we will have the CRM ready for them for the conjugation. Next example is one from peptides. This is a group in West Virginia, and Dr. Sen Kilich very kindly provided this information. This peptide 2 from the Pseudomonas outer membrane protein had previously been shown to as a KLH conjugate to induce protective antibodies. We made the CRM conjugates for them, and as you can see here, the antibody response to the peptide was much higher with the CRM than it was with KLH. The most likely reason for this is not necessarily, I would say, that the CRM is such a far superior carrier protein, but that the KLH is very difficult to characterize the KLH conjugates, And as I showed you with our analytical data, we could be quite sure that we made a good conjugate with the CRM. Whereas it's not impossible, but it's extremely difficult to characterize conjugates made with KLH. They did a challenge study, and consistent with the antibody response, the peptide CRM conjugate reduced the bacterial load. The dotted line with the arrow is the level of reduction, bacterial reduction with the reference whole cell vaccine. So the CRM conjugate was much more effective than the KLH and approached the level of the whole cell vaccine. The last section that I'm going to talk about is really not with our ready to conjugate proteins, but I thought was such an interesting area, and there's been probably the most publications that use our CRM. And I'll finish up with this section. So these are going to be chemical haptens. The idea is that you will make a hapten out of the drug, such as heroin, immunize with it. You'll make antibodies against the drug, and these antibodies will then bind up the drug and prevent it from reaching the brain. I'm not going to address the idea about whether this is a good idea, has any value or anything else. But there certainly is a lot of interest in it. We began working with Marco Provatoni at the University of Minnesota. He had previously been making KLH conjugates, and then he evaluated a series that he made with tetanus and CRM as well. First thing, he noticed that it was marked. He couldn't characterize at all the tetanus or the KLH conjugates, where he could easily characterize the CRM conjugate with mass spec. He immunized rats with either oxycodone KLH or oxycodone CRM, and had a much higher antibody response against the oxycodone with the CRM conjugate. And then he measured brain uptake in sort of a challenge study. And the CRM conjugate, the rats that had been immunized with CRM had much lower levels of oxycodone in the brain, so it achieved its mission. This last week, I received data from collaborators in Texas from Tom Costin's group. He's making fentanyl CRM conjugates. Again, previously he had been working with tetanus and KLH. The rats that received the control and then exposed to fentanyl went comatose and were insensitive in the tail flick assay. In contrast, the rats that received the fentanyl EcoCRM conjugate were happy and perky and were sensitive in the tail flick. So it was quite successful. Hopefully they'll be publishing this, and hopefully they provided me with a video, but it wasn't sufficient quality to show, but it's pretty interesting to see the difference between these. I will say the comatose rats apparently did revive. So I'm going to just finish up this seminar with a little bit about my personal mission in this field. Although we are a commercial company, my personal mission is to help make conjugate vaccines affordable for low-income countries. So our business model is a little bit different than many companies. We're what I call a not for much profit company, in that money is not our primary goal. We have no outside investors, and we have the motto, doing good while having fun and trying not to go broke. In our nearly 15 years, I'll say we've got the first two down. The third one is a constant struggle. To illustrate some of the struggle, in the last slide, I will have a cartoon model that shows you how we are achieving GMP CRM without having any investors and without being beholden to anyone. Because if we take other people's money, their goal will be to try to extract as much money as they can out of this product. And our goal is, in fact, to provide it for as little as we can while still trying not to go broke. So this is the story of GMP EcoCRM. It began when it starts, of course, we have no money. If you're familiar with GMP, even for phase 1, it is typically a multi-million-dollar undertaking. We began working with PATH. They provided us some funding for EcoCRM. The rest came out of our own funds. After we had developed it and we were looking for some funding for the GMP production, PATH, we worked out an arrangement where we gave PATH a royalty-free license for use in Gavi countries, which are the poorest of the poor countries. We also gave a license to a company in Seattle called InventPrize, which was founded and run by the former director of the Serum Institute of India that I mentioned earlier on. He's now retired, come to the US and started a company with a similar mission to ours of making affordable conjugate vaccines. We gave them, InventPrize, a license to use our CRM. In turn, PATH provided us funding to begin the scale up. Not to complete the entire GMP process, but at least to bring it to GMP. So they have funded the work that you saw in that slide where I showed the pre-engineering run. Sometime after in the year after we provided InventPrize a license, InventPrize received funding from the Gates Foundation to develop pneumococcal and Group B strep conjugate vaccines for Gavi countries. So now InventPrize had further funding to move the CRM into GMP production because they needed EcoCRM for these conjugate vaccines. So this is our process for generating GMP EcoCRM, and without any investors, and that in turn leads back to pulling money out of a hat. And that's how the magic trick that I've almost done with. So that winds up the seminar. Thank you very much. Thank you for spending the last most of an hour. Thank you to the GlycoNet. And all this time I've been sitting here with my pet rabbit, Miss Molly. She's listening with all ears. Thank you very much, and I'd be happy to take questions.
Great webinar. So I think we're about out of time today. So I'd just like to thank Andrew and everybody once again. Thanks very much, and hopefully we'll get to meet in person soon.
Yes.
Thanks, everybody.
Thank you.